screening for biostimulants. So they have some techniques to identify biostimulants, and they add biostimulants to the plant. So they have thousands of pots, and they want to see when do I have a reaction of my plant. It could be a growth promoting effect, it could be a change in color, etc. So our sensors, in this case it's two, in order to overcome occlusion, they're bound to these kind of gantries that move across the greenhouse, and here we do thousands of measurements per day and develop or deliver, let's say, information about the reaction of a plant towards a biostimulant. So that's very typical applications. We do it with biostimulants, fertilizers, crop protection industry. The same thing also, it could be hooked for breeding processes to tractors. The same thing, tractors drives over the field, we capture the plants which are below, process all the data, deliver this information to a scientist. So either it's tractor bound, but we have also a company from France which is totally autonomous and which are driving our sensors to the field. And this is now, let's say, the bridge, and this is to, let's say, the horticulture industry. We can also use this kind of irrigation system. You cannot see it, but here, this little box, that's actually the PlantEye. And it's a spraying boom which is moved across the greenhouse, and we just measure all the plants which are below and can also extract all these parameters. And the thing what we get out of this, if you measure one time, you get a piece of information, but if you measure obviously several times, you get also the dynamics, right? I mean, we saw before the talk from Eric. There we saw actually the end result, but actually what happened in between, right? And can we use that? And this is an example, like real data from a greenhouse. So it is completely, it's not cleaned. And what you see here, that's different days. So we have here 10 days. We have here in this case, this is the height. So from here to here, that's 10 millimeters. We're talking about this. So just to give you an impression about the precision of the system. And what you see here is an up and down. And that's not a measurement arrow, it is actually, that's why it weighs tomato, it's not written here, but it is actually the diurnal rhythm of the plant. So the leaves are moving all over the day. You could see it a little bit in your videos, but the plants are adjusting their leaves. And at midnight, and this is also species dependent, here these are peaking at midnight, but you have some also which peak at 3 o'clock, so, but you have always this up and down. And this up and down is really like a heartbeat. And all of these lines is actually representing one batch. By the way, that's young plants. That's what I said before, it's over 10 days. And these plants were exposed to different treatments. We're going to have a look actually how the different stresses are actually affecting my pattern. So here at the beginning, the 4 plants, or these 4 batches, it was just a control group. So all of them are pretty nice and synchrone. And then on this day we added a very moderate salt solution to these 3, except of that one here. You see this one is a little bit higher, but those guys within 24 hours are reacted to it. Again, here we're talking about 5 millimeters, 10 millimeters. So that is something which cannot be seen by human eyes, but the machine can immediately detect that. A change of growth, that means something is happening. Then, next day, here was the weekend, we didn't water the plant. Immediately the plant stopped growing. Tuck. And then Monday morning we supplied a really heavy stress, high salt treatment, and there we can really see that the plant has problems with its tuber, somehow is catching up. I mean, it is not rocket science, but what I would like to show you that by observing the pace that the plant is growing and the change, that means that something is not in order. Same thing with the same technology, we can also capture diseases. In this case, this is cabbage, and we're actually analyzing the effect of illness. It's a bacteria, it's Xanthomonas, and breeders, they breed for resistances against Xanthomonas. And also here we use PlantEye. Here we use indeed some spectrum which we can't see by human eyes, and we can discover all the regions which are affected by the disease, and we can quantify that, really give a number. It's not something like I give it a 5 or maybe a 6 out of 10, it's a real number. And this number is also corrected for a perspective, because we have 3D information. So we don't care whether the leaf is like that or like that, we can correct for this, right? And if you can compare this data with the information we get for the breeders, we get a 0.9 correlation. That means you can really automate what breeders are doing every day in the greenhouses on the field. So these are two examples. Again, that's growth. That's something, what I just said, like growth is obviously an integrated parameter. There's many different building boxes which lead to growth, and we cannot tell what exactly is happening, but we know if growth is changed, that something is happening. And here we have some numbers about diseases, but how can we then make this usable actually for growers? And this is something actually what we did together at the grower. In this case, it's an ornamental plant. And what you see here, each of these blocks is actually a batch of plants. And the grower knows exactly his perfect growth rate from experiments he did before. And what we did here is that we can also specify a range, a certain range. And if we are within this range, that means it is okay. And you see these shades of green also show different growth rates. By the way, that's per day. You see these dark ones. Here the plants are growing a little bit slower than it should be, but still it is within the range. But on those spots, this is really red, so here we are out of the range, and that means here is a problem. Right? We're using the growth data, we visualize that. We cannot tell what's happening, but the grower can at least have an insight into his farm. I guess for all these indoor farms that are operated worldwide, what Pavlos was just presenting, this kind of things might be of great interest. So, another application, you see this big machine, it's a lettuce harvester. The lettuce harvester, it's a dinosaur, this is our sensor, and the entire dinosaur is controlled by our sensor, right? So these lettuce, normally they're cut by humans all day long, worldwide. The problem is there's less and less people who want to do this heavy job. So the growers are facing the problems of finding actually the labor to do so. So we try to do exactly the same thing what humans does. So the sensors are actually phenotyping the crops. And the first decision we do is, should we harvest it, yes or not? And there obviously there's some criteria of the supermarket. It could be this height, it could be the diseases, illnesses, etc. And then if we have this first information package, our sensor will make a decision, or our algorithms, which is running in the sensor, will make the decision, okay, we can harvest. That's the first step. But then we have also to guide the robots and compute exactly the position of it, and also compute the perfect cutting height. The thing is, if you cut too high, the complete product is destroyed. If you cut too low, you have these leaves which are close to the soil, which are dirty, which might have infection, etc. So in this case, you need a post-processing. So we don't really save labor. You save labor maybe the guys cutting, but the guys in the back of the truck taking off the leaves, that's something you don't want to have, right? So again, this entire process is done by our sensors. So you see these are, in this case it's romaine heart. We scan these plants, we digitalize these plants, we make first a decision, harvest yes or not, and then we compute the parameters required by a machine and to drive these machines. Also here, Pavlos also mentioned it in your talk, robotization. I think the entire industry will be automated in future, and for that, at the beginning, you need also sensors which acquire in high precision and also under any conditions, right? Agriculture is a lot of dirt, there's light, there's wind. This is really not easy actually capturing plants under those conditions. So now the last slides, a little bit more of concentration. Now it's getting what we did together with Fluence. So I think we talked to Fluence beginning of the year, because we said, we see all this lighting industry, we have more and more clients coming from the indoor production, and we see that we can manipulate a lot of light. And when we go to conferences, we see actually the results, or we can manipulate the light composition, and then we see the results after 6 weeks, after 7 weeks, etc. But what we wanted, or what we were actually interested in, how quick are these processes, right? I mean, is it something which takes weeks, or is it something which happens within hours, right? And that was a bit the idea, so we collaborated with Fluent. So unfortunately you cannot see them, but up there is the phytophile lamps. And I guess you guys are all familiar with the phytophile lamps. It's these 5 wavelengths, so it's quite nice. You can exactly structure the light composition and don't change actually the intensity. So we made several experiments, and it's similar to what you did, Eric. However, it's not. The purpose is not really to have deep or to have like a rocket science there. And what you did is on a completely different level. For us, it was just interesting. How quickly do plants respond to that, a change in light, and can we capture that with our systems? So, first result, it is again, this is days, again I think it's 10 days or so, yeah, 10 days. In this case, again, it's height, so you see again the pattern, up and down, and what we did here, we had, by the way, we really separated also, we had this curtains, yeah, because, yeah, we had those. So,
we had to leave it.
These plants here, they were had exactly the same light treatment, so that was blue together with red, and we wanted to add then far red and see how this is actually influencing the growth. We knew, I mean, it's not rocket science, but we knew if you add far red, there will be an elongation, right? So we want to see how quick does it go. So again, plant grow, and then we swapped. So we added here far red, by the way, thanks to Sebastian, because he provided us the light recipes. And we continue to measure, and you see there's a reaction after 24 hours. You can see the plants treated with far red, they grow quicker, elongation. So yeah, that's cool. There's something happening. It goes, the reaction is quite fast. So actually we're quite happy to that we change the light composition, and actually almost in real time you can see the reaction on plants. So that was the first experiment we did. The second experiment is that we looked actually at green light and blue light. So how would that actually affect our growth dynamics? And again, the plant here, they were subjected to the same light treatment. It was green and red. So both plants here, green and red. In this case, it was biomass, by the way. And then we changed this one here. We added blue light. This one here was kept still at green light. And here the reaction was much quicker. I mean, the difference here is a bit that these plants just continue to grow. This one here was just slower in growth, right? But the effect was almost, within 12 hours. We looked also at the color. That was also something what we can capture. Again, this is the time. Here, in this case, it's called machines, they cannot tell whether it's red or light red, so we actually transform a color into a number. And very roughly, red here is 0 and green is 90 degrees, and in between we have all these different shades of color. And that's exactly the scale what you have here. So we put the plants into this, into the experiment. The plants were coming from a grower, so they had somehow to adapt a little before, because they were growing outside, they were transported, so somehow they had to adapt. But then they are pretty well synchronized. And then again, we swapped those and added, instead of the green, we supplied it with the green with the blue light, and we could see a change in color. And this change was really almost immediately. I went back also today, I thought, that's maybe not possible, that we have such a fast change, but it is indeed something which is really physiological there, and we had also some measurement targets in there, and they were always